Okay, so good evening, everyone. We will begin with our discussion for today. So, before starting the exam, because people are yet to join it, so I'll begin with a recap of the topics that have not been included as part of this exam. So, I'll just quickly start off with a brushing up of those topics, and then once we have people on board, then we can proceed with the exam. So just a moment, I'll switch off my video and share my screen. Okay, so if you remember our last discussion and last revision and the topic that has been included as part of this first examination has been Sanger sequencing. So we had discussed NGS methods, Sanger sequencing, etc. And then what we had begun next. Just a moment. Yeah. So then we began our discussion on next generation sequencing techniques. So if you remember, we had first started off with a clear distinction between NGS and Sanger sequencing method. So just for some foundational difference and just to brush up our basics regarding the same. So Sanger sequencing, if you remember, involves individual nucleotides, not nucleotides, but individual oligonucleotide sequences or oligonucleotides being sequenced. And while its initial idea involved the use of radioactive isotopes, it was later allowed to evolve. And after advancement in technology, fluorescent probes started getting used so as to eliminate the health risk and the life risk that tags along with this as well. So just give me a minute. Let me reshare it. Okay, so coming back to the presentation, let me reshare my screen. Okay, so we were discussing the attributes of next generation sequencing. So what is the idea of next generation sequencing and how is it in contrast to Sanger? So NGS involves the rapid sequencing of very large genomes from many different types of organisms. Secondly, oh, let me just put this into slideshow mode. Yes. So rapid and deep sequencing of target regions, followed by the utilization of RNA-seq to discover novel RNA variants and splice sites or quantify them for gene expression analysis. Analyze epigenetic factors like genome by DNA methylation, DNA protein interactions, etc. And also to carry out this sequencing so as to figure out some rare somatic variants and obviously utilize that for clinical translation so that we can we can detect, we can find biomarkers, we can understand disease progression, etc. Study the human microbiome, and finally identify novel pathogens. So that is the whole idea. Now, if you look at the overall workflow and the contrast between Sanger and NGS, you will see that there are um, two sets which are obviously common between the two. One is your template that you want to sequence versus NGS is where you have millions of templates versus when you look at Sanger, there's only one single template which is present. There is extension, arrangement, and then detection based on the length of the nucleotide. And then you get these sharp peaks. You do not expect shoulders. You want clean peaks in this case so that there is no discrepancy as to what nucleotide is present at that site. So basically, the sharper your peak, the more reliable your result is, the more accurate that is, and you will have more confidence in your data then. And if you look in contrast, when we talk about NGS, then in NGS you have arrangement over a solid support. Most commonly, we'll be discussing this, but most commonly it's a well that is meant for allowing the sequencing reaction to proceed, followed by extension from nucleotides, which are repeated over 100 times, the detection by incident lasers, and finally you detect them as these fluorescent signals, which you can see there's another step involved, which is signal amplification, so that the signal you get is more detectable and more reliable. And how that happens is also something we'll be revising very soon. Now, when we talk about next generation sequencing or NGS methods, some key concepts involve the fact that there are four main steps. First, sample extraction. Second, library preparation. Third, sequencing. And fourth, analysis. So, second generation sequencing, like we have already previously studied, it involves some of these key steps. First involves DNA fragmentation, then there is preparation of a library. So, what you expect in that case is that either there can be cloning of a library with using bacterial systems so that there is more amplification, or you can perform adapter ligation as well. Then there can be cyclic sequencing, which is then read using electrophoresis. Or there is generation of a polony array, which are PCR colonies. And then there, cyclic array sequencing is carried out, which allows you to get results, high throughput results of approximately 10 to the power 6 reads per array. So, with high throughut shotgun Sanger sequencing, genomic DNA is fragmented, then cloned to a plasmid vector and used to transform E. coli. For each sequencing reaction, a single bacterial colony is picked and plasmid DNA gets isolated. Then, cyclic reaction takes place within a microliter scale volume, generating a ladder of ddNTP terminated dye-labeled products, which are then subjected to high resolution electrophoretic separation within one of 96 or 384 capillaries in one run of a sequencing instrument. Now, when these fragments they pass a detector, there is an emission spectrum which generates a sequencing trace. Now, in shotgun sequencing with cyclic array methods, common adapter ligation takes place, which results in the generation of spatially mobilized PCR colonies called as polonies. They contain many copies of a single shotgun library fragment, which are all tethered to a planar array. And then you can use imaging based detection to figure out which fluorescent label is present. And when this is done iteratively, then it allows you to build up a contiguous sequencing read for each array feature, and then you can interpret your results. So that's what we see here. Then second generation is the concept or the terminology or the keyword of second generation usually refers to the various implementations of cyclic array sequencing that have been realized in a commercial product. For example, there can be Solexa technology, which is used by Illumina, or there can be, or otherwise 454 sequencing by Roche, the pollinator, helioscope, single molecule sequencer technology, etc. Now, what do we exactly mean by this terminology, which is cyclic array sequencing? It refers to the sequencing of a dense array of DNA features where there are iterative cycles of sequencing of the same region using imaging based and then detection of the data or collecting the data using imaging based detection strategies. So here's what we see. The first step in this strategy is the reliance on emulsion PCR, where the idea is that you want to amplify your clonal sequencing features. So if we just summarize it briefly, since this has already been discussed, you have adaptive flanked shotgun library, which is generated, and here it has been shown as gold and turquoise adapters, which are which have been ligated to the different templates or the short fragments that have been generated as part of the shotgun approach. And then it undergoes PCR amplification, and that amplification takes place within a small bubble or a small emulsion, which allows for which basically constricts the region within which amplification can take place. So apart from your template, what else do you put in it? You put in your beads with the immobilized adapters, and then within that, emulsion PCR is carried out. Now, just give me a moment again. Yeah. So, how are these emulsions or these droplets generated? Basically, it involves a water and oil emulsion generation. So there's agitation and droplet generation within which, within each of these droplets, this PCR reaction proceeds. And one of the PCR primers is tethered to the surface of this micron scale bead that we already mentioned. And because you have limited or controlled template concentration as well, it results in most of these bead containing compartments either having 0 or 1 template molecule present. In productive emulsion compartments, PCR amplicons would be after PCR, the amplicons would be captured on the bead surface. And once you break the emulsion, those beads that are bearing the amplification products, they can be selected out or filtered from the rest of the population. And what you expect is that within such droplets or within such bubbles, which allowed for this proper reaction to proceed, you will have beads that carry clonal amplification of your um, target fragment, and this is basically enriching your signal. You are amplifying the amount of template that can be used for the sequencing reaction from that point onwards. So the beads basically bear amplification products, and these correspond to the amplification of one single molecule from the template library. So you'll have hundreds and thousands of such sequencing reads which have been amplified on the surface of these beads. So following this, the next step is library preparation, which is accomplished by random DNA fragmentation followed by in vitro ligation of common adapter sequences, the generation of clonally clustered amplicons to serve as sequencing features. Can be achieved by different approaches, including in situ Bolognese, emulsion PCR, or bridge PCR. So, this bridge PCR strategy is what Illumina has extensively standardized and what we have already discussed. You also have the video link for that present on the slide. So, Alexa relies on this bridge PCR strategy as well to amplify clonal sequencing features. So basically, the adapter flanked shotgun library, which we saw in the previous strategy as well, gets PCR amplified, but both of the primers, they are present on the surface of a solid support, most commonly known as the flow cell, which is attached at the 5' ends by some flexible linkers. So uh, certainly when you want to immobilize a piece of DNA, some sort of a linker, some sort of a well, in layman terms, some sort of a glue must be present, which allows the mobilization or the positioning of these probes on the surface of the flow cell. As a consequence, amplification products originating from any given member of the template library remain locally tethered near the point of the origin. At the conclusion of the PCR, each clonal cluster contains thousands of copies of a single member of the template library. Accurate measurement of the concentration is critical to maximize the cluster density while simultaneously avoiding overcrowding. Yeah. So now what happens if we zoom in a little bit more into the reaction, sequencing reaction that's actually taking place? So on the Illumina platform, the bridge PCR you see is quite unconventional in relying on alternating cycles of extension with BST polymerase, denaturation with formamide. So let's focus a little bit here. So the clusters that you get, they consist on thousands of approximately 1,000 clonal amplicons, which can be amplified to certain locations within each of the 8 independent lanes present on a flow cell. Now, after generation of the cluster, the amplicons becomes single-stranded or they are linearized and a sequencing primer gets hybridized. If you look here, that's what you see. This is your linear PCR amplified amplicon, and to it is hybridized a sequencing primer that flanks your region of interest that you wish to sequence in more depth. Now, each cycle of this interrogation consists of a single base extension. Every time there is sequence interrogation, or every time the sequence is put under the microscope to be decoded, there is a single base extension with a modified DNA polymerase, and you of course have all four nucleotides available. So whichever gets incorporated will emit will be fluorescently tagged with a different marker, and based on which one gets incorporated, you will have the release of that marker that you can then detect. So image array, remove label and terminator, add fresh nucleotide. So basically what you do is after the incorporation of the first nucleotide, you image the array, then you remove the label and the terminator, then you again add a fresh bout of nucleotides and the polymerase. The next base gets incorporated. So your first signal was blue because it incorporated thymine. Your second signal is green because it has incorporated cytosine, and so on and so forth. Now, after this, these nucleotides they also can also be modified in two different ways. They are often also referred to as reversible terminators. How does that, or what does that exactly imply? So, terminators, in the sense that they can terminate the sequencing strategy at some point, and they are reversible because there is a chemically cleavable moiety that can be removed, or that can be cut off in presence of a certain stimulus. So what it does is that the chemically cleavable moiety allows only single base incorporation to occur in each cycle, and along with it, one of the four fluorescent labels also can be detected, and that corresponds to the identity of the nucleotide which has gotten incorporated. Yeah. Now, after single base extension is carried out, an image is acquired at in all the four channels. Chemical cleavage of both groups sets up for the next cycle. Next is emulsion PCR. So 454 Roche, solid, gene reader, and ion torrent. So emulsion PCR, as we have already discussed, involves the formation of these droplets within which you have your bead that has all of the adapters ligated. And what you expect is on bead amplification to proceed in this manner as shown here. So like you can see, the red sort of cylinders, they represent tiny little probes that have been localized on this surface, and from that you can see that there is sequencing amplification which can begin or can be initiated from those primers, and then you will have a final product that has 100 200 million beads which have thousands of the bound template that you can take forward for sequencing. So yeah, so this is how PCR is carried out. So these are also called as template amplification strategies. The other possibility, which we have also discussed once before, is the solid phase bridge amplification strategy, which is utilized by Illumina. So there, fragmented DNA gets ligated to adapter sequences. The initial part is of course the same. Then it is bound to a primer, which is immobilized on a solid support like a patterned flow cell. The other end, or the free end, then interacts with other nearby primers that allow it to form this sort of bridge structure. Then PCR can be utilized to create a second strand from these immobilized primers, and unbound DNA gets removed. You have these sort of bridges that are now formed, which can again denature, linearize, and form uh, longer sequences that can form intact sequences which can be taken forward. So that's pretty much it. So if you think about it, there's not that much different. It's just that the surface is variable between these two strategies. And second, the first one involves the generation of droplets. The second one does not. When we talked about other strategies for template amplification, so if you remember in solid phase template walking, The fragmented DNA gets ligated to adapters. Again, the initial part is the same, and then it is attached to a solid support, like I previously mentioned. So then PCR can be utilized to generate a second strand, which is partially denatured, allowing the free end of the original template to drift and bind to the other nearby primer sequence. Reverse primers are used to initiate strand displacement to generate hmm, additional free templates, each of which can then bind to a new primer in DNA nanoball generation, which is the second strategy. Okay, let me just focus with the image here. So if you see here, you have binding of the template initially where your free DNA template hybridizes to those red colored bound primers like we already talked about in the previous emulsion PCR strategy as well. And then you have amplification of the second strand because these are sort of universal primers and they can bind to certain defined regions of these templates. Then second part shows you primer walking, whereby dsDNA gets partially denatured, allowing the free end to hybridize to a nearby primer. Then there is template regeneration, whereby the bound template gets amplified and free DNA templates can be regenerated. There is cluster generation so that there can be effective signal amplification as well. So yeah, so that is the solid phase template walking, which is a strategy that is used by a solid sequencing system. The fourth one is in solution DNA nanoball generation. If you remember, we had seen that there is formation of sort of concatemers like structures. So you have adapter ligation to either end of a DNA template, again shown here in red, followed by circularization of the template. Now this forms a covalently closed circle. Next, these templates are cleaved downstream of your original adapter or adapter 1, if I may say so, the red colored one, following which you will have the incorporation of adapter 2, 3, and 4 in an iterative manner, circularization, cleavage, generating a circular template with 4 different adapters, as you can see here. Now, these can then be amplified further by the rolling circle mode of amplification, where there is a where sort of concatemers can be formed, which independently, if cleaved, can also form potentially also formed covalently closed plasmid-like structures. But however, it does not happen. So instead, there is a constant synthesis in a in a loop-like manner, and these intermolecular interactions between these entities is what keeps the nanoballs cohesive and separate in solution. Then hybridization, the nanoballs get immobilized on a pattern flow cell. Then the process is repeated for the remaining 2 adapters. Final product is a circular template where you have all 4 of these adapters, which are separated by some region of the template sequence. Now, library molecules undergo a rolling circle amplification step, generating large concatemers called as DNA nanoballs, which can then be deposited on a flow cell, and that allows detection also. Now, again, one might ask then, if that is what you eventually are going to do, what is the need for going through all of these previous steps as well? Well, basically, the core idea, the crux of this whole thing remains the same, where we want to enrich for our template as far as possible and as much as possible. And for that, if you have repetitions of your sequence of interest in tandem within these nanoballs or within these loops, then that will enrich for your DNA and enhance it basically. Hmm. So this is a little more in-depth insight into the solid sequencing strategy. So if you remember that after cluster generation or the deposition onto the slide, we move on to a second set of strategies which are different from, um, we move on to a different discussion at all. So if you go back here, yeah, so these were template amplification strategies. We talked about 4 of them. Then we move on to the methods of actual sequencing after your library preparation has been completed. So the first one involves the use of sequencing by ligation strategies, where it is very heavily reliant upon complementarity and upon hybridization and upon ligation between these as well, between broken portions of DNA or separate portions of DNA. So fragments that can often be sequenced by ligation-driven strategies, which whereby a fluorophore labeled two-base encoded probe, let's say for example AT shown here in blue, is followed by degenerate or universal bases shown in pink, are added to the DNA library. Now, what you expect is that the two-base probe can be ligated onto an anchor shown here in purple in this color, and the slide is then imaged to identify the first two bases in each fragment. So first you have ligation of the probe to an anchor, which is adapter complementary. That is why adding the adapter is necessary so that we can have something to start off with. And yeah, the unextended strands get capped by unlabeled probes or phosphatase to maintain cell cycle synchronization. Finally, the terminal degenerate bases and the fluorophore are cleaved off the probe, and that leaves a 5 base pair extended fragment. The process is repeated 10 times until 2 out of every 5 bases can be identified. The entire strand can be reset. So, effectively, what you are seeing is that you have a 2 base probe, then you have a set of degenerate bases, which are tagged with a fluorochrome. So, when your degenerate base containing probe is able to find complementarity, then the fluorophore gets cleaved along with several bases, revealing an acceptor 5' phosphate, which can then get ligated to a 3' OH end as per the availability. And then there can be extension of the probe. So what you basically get is a disturbed, sort of disrupted sequence where you have two bases which are known because they have come from your probe, and they're interrupted by these pink colored sequences which have come from your degenerate regions. So how do you figure out what are the missed out sequences from these degenerate portions? So to compensate for the same, then the next round of iterative sequencing, you change the anchor and you provide the N+2 offset. And as a result, what will happen is that you'll see a shift of two bases, which will compensate for whichever nucleotides were already present there. For example, if you see here, GC was present here, that we already know. So if your anchor now carries this offset, then it is able to find complementarity here itself, and yeah, and then from that point onward, you'll have two bases again which are known. So effectively, you now know the third base of each subsection, and you can again repeat this with N+3, and then you will have all of the bases from your N+4 N and 4 sort of distribution. So yeah, the process is repeated 10 times until 2 out of every 5 base pairs is actually identified. At this point, the entire strand can be reset by removing all of these probes, and the process of binding, probe binding, ligation, imaging, and cleavage gets repeated again, each with an N+1, N+2, N+3, or N+4 offset anchor. Next, we have complete genomics, whereby this is a different strategy, which involves the use of a CPAL named approach, which is combinatorial probe anchor ligation approach. After DNA nanoball deposition, after DNA nanoball deposition, an anchor which is complementary to one of the four adapters. Yeah, one of the four adapter sequences and a fluorophore labeled probe are bound to each nanoball. The probe is degenerated all but the first position. So the first position is known, the rest of the probe is actually degenerate. So there's a 1 out of 4 chance for each of the probes to actually bind. The anchor and the probe are then ligated into position and imaged to identify the first base on either the 3' side or the 5' side of the anchor. Next, the probe anchor complex is removed and the process once again begins with the same anchor but a different probe with the known base at the n+1 position. So again, you put in an offset of like a single base, and then you'll have n+1, which is known, and then you'll just end up with a few more bases not known. So another round of this hybridization is taking place, and this time using anchors with 5 base pairs offset, identifying additional 5 bases on either side of the anchor. Finally, this whole process is repeated for each of the remaining 3 adapter sequences in the nanoball, generating 100 paired end reads. Um, Cyclic reversible termination approaches are the next subsection. So we talked about sequencing by ligation. There were two subcategories under the same as well. Next, we talk about sequencing by synthesis, which is used by Illumina, the most common, the most abundantly used, and very relevant in today's time. So that's the strategy we discussed next. So we talked about how after solid phase template enrichment, there's a mixture of primers, DNA pol, and modified nucleotides that are added to the flow cell. Now these nucleotides are blocked by a 3' O azidomethyl group and is labeled with a base-specific cleavable fluorophore. And during each cycle, fragments in this cluster will accommodate just one nucleotide as the block 3' group will, prevent any additional incorporations. Yeah. After base incorporation, unincorporated bases get washed away and the slide is imaged by TOR. Then it basically allows you to see which base got incorporated. To simply put, there are 3 steps. First one is that you have to add or DNA polymerase will allow for the incorporation of the nucleotide, which is which shows complementarity with your template, and then that gets incorporated and releases a definitive signal, red, green, yellow, or blue, following which the slides are imaged with either 2 or 4 laser channels. Each cluster emits a color corresponding to the base incorporated during this cycle. Just give me a minute. Yeah. The second strategy, which is used by Gene Reader, owned by Qiagen, involves nucleotide addition. Just a minute. Yeah. So this involves nucleotide addition, where you have 3 or 4 labeled terminally block nucleotides and unlabeled block nucleotides. They um, hybridized to complementary bases, and each bead on a slide can incorporate a different base. Then you image it using 4 laser channels, and it emits a color corresponding to the base which got incorporated, but only the labeled bases will actually emit a signal. And third is that there is cleavage of the fluorophores and they are washed from the flow cell, and the 3' OH group gets regenerated. A new cycle begins with the addition of new nucleotides. I'm not going over the legend, we've already discussed it, but this sort of ended our first discussion regarding this topic. Next, we moved on with sequencing by synthesis, single nucleotide addition approaches. So we talked about sequencing by ligation methods. We talked about sequencing by synthesis methods. And next, you have sequencing dependent on single nucleotide addition strategies. So again, you have the 454 pyro sequencing method. So the initial part is it shows you a recap of the emulsion PCR strategy we have already talked about. Now, after template enrichment in this bead-based strategy, beads get added onto a microtiter plate along with primers and different beads that contain an enzyme cocktail. Now, during the first cycle, a single nucleotide species gets added to the plate, and each complementary base is incorporated onto a newly synthesized strand by a DNA polymerase. So, what do you see here? You see that during the first cycle, there is a single nucleotide species that gets added. So, for example, here shown in red, which gets added, and yeah, and this is performed by DNA polymerase. The byproduct of this reaction is a PPI molecule. Polymerase here is shown in blue, and as a result of incorporation of the base, pyrophosphate is removed and or is released, and this molecule, along with ATP sulfurylase transformed APS into ATP. So APS is taken up as a substrate and adenosine 5' phosphosulfate and is converted into ATP, adenosine triphosphate, which in turn is a cofactor for the conversion of luciferin to oxyluciferin. So basically what you see here, you see that when there is base incorporation, PPI is released. When PPI is released, it converts APS to ATP, and when ATP is present, it acts as a cofactor of um, it acts as a cofactor for conversion of luciferin to oxyluciferin by the enzyme luciferase, and the byproduct for it or the readout for it is like yeah, otherwise also known as luminescence. Now, finally, apirase is also used so as to get rid of any unincorporated bases so that you do not get non-specific off target results. And the next base gets added to the wells. Each burst of light, which is detected as like a single charge coupled device or CCD camera, can be attributed to the incorporation of one or more bases at a particular bead. Oh, excuse me. I accidentally minimized it. So Iron Torrent is for Thermo Fisher, is hosted is a proprietary material of Thermo Fisher, whereby basically what they do is that after bead-based template enrichment, like we have already discussed, beads get carefully added into a microtiter plate where one bead is present in one reaction well. Then you add your nucleotide species one at a time and then perform a standard elongation reaction, which occurs naturally because of the presence of the enzyme. And as each base gets incorporated, a single H plus ion is generated as a byproduct. So every time base incorporation occurs, H plus ion is released, which results in a 0.02 change in pH detected by an integrated complementary metal oxide semiconductor and an ion sensitive field effect transistor or effect device. And after this introduction of a single nucleotide species, you can wash away, all of those debris and all of those unbound bases so that that does not skew or bias your results. And then the next is at short read versus long read technologies or techniques. So short read technologies, they actually involve a fixed runtime, increased time to results, and an ability to identify the workflow. There are multiple practical complexities that are associated with handling and storing large amounts or volumes of sequence data. Long read technologies are more advantageous in this question in this aspect because there's more real-time data acquisition. There is much more rapid turnabout with immediate access to results. It enriches single targets during sequencing with no additional sample prep using adaptive sampling. Then limited flexibility of short read sequencing technologies, which is because of sample batching that is often required for optimal efficiency, potentially leads to very long turnabout times. Benchtop devices confine sequencing to very like sort of centralized locations, whereas for long read sequencing, it is much more scalable, much more flexible, scaled to suit the throughput needs. And you can decentralize sequencing, no sample batching is required. The key difference, of course, which is the reason these names are attributed to each of these sequencing strategies, is that short read sequencing techniques, they obviously generate much shorter read length, whereas for long read sequencing technologies, we have much larger read lengths, which can go up to 4 MB or more. So the information you get out of it, of course, is different. And what do we mean by the information we get out of it? It's basically that the depth of the sequencing and the length of your sequencing read, which is generated, that is so different that the interpretation you can make from that data. But technically speaking, once you have stitched your sequencing reads back to the reference genome, which is what we learned in our last class when we discussed star aligner and the tool which is used for that purpose, while that allows essential stitching back of your sequences, it becomes a little unreliable to use these techniques interchangeably. Why so? When we talk about short read sequencing technologies, they are not actually taking large regions into consideration. Think of it like a long thread. And when you chop that thread into smaller pieces and then you try to tie it back together, it will not be the same, right? So shorter reads, while they are useful for better depth and for more basically sort of zooming into the information that is present in the genome, it's not very useful in the context of having to um, when you want to interpret structural aberrations or you want to see very large alterations, like there are gene amplifications or any these sort of changes. So mainly for structural variants, it's not very advisable to opt for such short read technologies and instead look at instead take a bigger snapshot and sort of like a panoramic view at the same time and then interpret results from there. So based on what your target is, what your hypothesis is, you choose the appropriate strategy. They both have their pros and cons, and the availability of both is subject to what you want to experimentally um, eventually figure out. So that is the whole idea. Now, for these sequencing approaches, there are different types of templates which are present. Let me just quickly revise this particular template because it was so unique. If you remember, this is the smart cell, which is at the very heart of smart sequencing, which contains millions of tiny wells called as zero mode waveguides. Single molecules of DNA get immobilized in these wells, and polymerase incorporates each nucleotide. So they're sort of like a double-headed hairpin-like structure. So you have your DNA fragment, and at its end, you have these hairpin adapters, which are ligated to both of these ends. And you use these sequencing primers, which initiate polymerase-driven sequencing from these sites, and then it keeps on continuing in this manner, and that allows for circular sequencing, very similar to what we had discussed for DNA nanoball generation, except that it's not a double-stranded DNA, but instead a single-stranded DNA like structure where a central portion has very high complementarity because they are originally part of the same fragment. So, yeah. So, basically, template fragments are processed and ligated to hairpin adapters at each end, which results in this sort of circular DNA molecule with constant single-stranded DNA regions at each end with the double-stranded DNA template present in the middle. And yeah, and the resulting smart bell template, which we have already discussed, undergoes a size selection protocol in which the fragments that are too large or too small get removed to ensure efficient sequencing. Primers and an efficient FAJ29 DNA polymerase are attached to the ssDNA regions of the smart bell. The prepared library is then added to zero mode waveguide smart cell where sequencing can take place. And you have this mixture of labeled nucleotides. And yeah, during incorporation, the nucleotide momentarily pauses through the activity of the polymerase at the bottom of the ZMW, which is a zero mode waveguide well, which is the hexagonal well you can say on the left, very similar to what we have previously discussed about the flow cell. And all four DNTPs, they are labeled and available for incorporation. And when the nucleotide is incorporated by the polymerase, the camera records the type of emitted light or the color of the emitted light, and based on that, you can tell which base actually got incorporated, and you will get more idea about it. Then you have real time and synthetic long read sequencing approaches. So yeah. So for synthetic long read sequencing platforms, you have this Illumina strategy once again. So you have genomic DNA templates that are fragmented to approximately 10 kb long pieces. Now think about it, 10 kb is still pretty long. And they are then partitioned into a microwriter plate with around 3,000 templates in one well. And within the plate, each fragment is then sheared. Matlab, it undergoes even further subdivision uh, to approximately 100 kilobases into micelles, which are also called as gems, along with gel beads containing adapter and barcode sequences. Now, what exactly do we mean by these gems? These gems, they contain around 0.3x copies of the genome. That is around one third of the genome. Yeah, they contain around one third of the genome and one unique barcode out of 750,000. Then within each gem, the gel bead it sort of dissolves and smaller fragments of DNA get amplified from original large fragments, each with a barcode identifying the source gem. After sequencing, the reads are aligned and linked together and they form a series of anchored fragments across a span of 50 kilobases. Now, this does not attempt to get full end-to-end coverage, but instead what you see, find a single gem that has for which the reads are dispersed across the original DNA fragment. And overall, the coverage that you get is derived from multiple gems with dispersed but linked reads. So linked reads basically imply that they are derived from the same gem derived from the long fragment, so they are considered to be linked. Reads get dispersed along the long fragment, and no gem achieves complete coverage. And stacking of the linked reads from the same loci achieves continuous coverage. So that's what we see here. Uh, I think Yeah, I'll end here. Then we had begun our discussion on sequencing the genome, epigenome, and the transcriptome, and we understood the contrast between whole genome, whole exome, and uh, yeah, the resequencing approach as well. So, target region sequencing or TRS. So basically, what you see, whole genome sequencing, as the name suggests, examines all genes including non-coding DNA as well at a sequencing depth, which is usually lower than what you would opt for when you're performing WES. Why? A, because the sample size is, of course, much larger. And secondly, because you have your objectives in this case are much more different. You want a more holistic view of the entire genome of that particular organism. But in contrast, for whole exome sequencing, the usual objectives are slightly different. But while its accuracy is low, its coverage is very high, and therefore that attributes the immense cost that goes into these sequencing techniques, which involve whole genome sequencing. And additionally, because your template size is so large and the amount of data which gets generated is also so immense, the depth usually for such cases is also much lower. When we say lower, what do we mean? It is approximately 30x or 30 to 40x approximately. And therefore, the depth there is not very high, but the coverage is much larger. larger. Now, if your objective is such that you want to opt for more depth into smaller regions and your objective involves only the coding, only focusing on the coding region of the genome, then the more strategically more sensible approach to opt for is whole exome sequencing, where, of course, when we talk about the exome, you are eliminating completely every other region of the genome except for the coding sequence, which is what composes the exome. You are fragmenting it, performing shotgun sequencing, etc., and you are utilizing only the exome part of the organism's genome and sequencing that. A, that reduces the computational power required. B, that reduces the amount of template that has to undergo the sequencing protocol. C, it gives you much better depth because smaller regions are can now be sequenced at a much like with more accuracy and by increasing the depth of sequencing. And fourth, it is very, very cost effective when you compare it to the expense you have to bear with whole genome sequencing. So these advantages make it a very useful and a different adaptation compared to whole genome sequencing. But again, you miss out on a lot of information as well. You are not, you cannot confidently conclude anything about the non-coding regions of the genome, and many a times many a times it's often very important to make sure that uh, you do not leave these less explored areas as it is, because it's necessary to understand regulation of the gene, not only the coding sequence of it, because that's not going to give you a complete, because that's not absolute by itself. Next, I think, yeah, we had moved on to exome, key steps in sequencing protocol, and expected outcomes, etc. So that part I will let it be for now. And instead, what I will shift the focus now onto are the questions that I had prepared for today's class, but unfortunately, nobody decided to join. And while that is very, very disheartening, the show must go on, and so I will take you guys through the questions so that you don't miss out on what was originally intended for you. So just give me a moment. Let me focus it. Yeah. So this was a 25 mark. I'll not upload this video now so that I can share this link over email and people can attempt it at their own time. Um, so, hey, I don't want to shift focus here. Yeah. So there were 25 questions for everybody to attempt. So let us go over them one by one. So which modification could have invalidated the conclusion of the Hershey-Chase experiment? If you remember, we had discussed this in our first class when we discussed the DNA, the discovery of DNA and all the experimental validation that was used for that purpose, using DNAs instead of RNAs after infection, using P32 labeled RNA instead of DNA, using S35 labeled capsid proteins, or any other option. So basically, the correct answer in this case is using P32 labeled RNA. Why so? Because RNA would completely skew these results. RNA also has a phosphodiester backbone into which P32 could have gotten incorporated, and therefore radioactivity would not have been the radioactive isotope would not have been able to distinguish between DNA and RNA and could have completely messed up the conclusion of the experiment. So the correct option in this case is P32 labeled RNA. Second, for Avery's experiment, if you remember Avery, MacLeod, and McCarthy, they had also performed a set of experiments with live encapsulated and heat-killed streptococcus, and from there they had figured out that there was some component of the cell which eventually turned out to be the DNA that was able to transform these heat-killed bacteria into virulent strains, and that of course, eventually turned out to be the DNA. So while this was okay, Why was there an initial skepticism? Yeah, that's what we've been asked. So, A, because DNA was thought to be structurally unstable, proteins were believed to carry genetic complexity, RNA contamination was suspected, or bacteria lacked a nucleus. It was actually because proteins were believed to carry genetic complexity, and um, for the first time, some other some contrasting evidence had been shown, and therefore initial skepticism was uh, was what Avery's experiment was followed by. Then Shargov's ratios, what do they fail to explain? If you remember Shargov's rules or Shargov's ratios, while his rules were have been transformative in the recent past, initially they failed to explain a few details. Um, what are those? If you remember the rules, usually what we consider the brief or the summary of those rules are that A equals T and G equals C. So, based on that principle, DNA complementarity and DNA structure and the stability of the entire structure, everything is reliant upon that simple rule. But um, which of these following things does this ratio actually not even address? The specificity of base pairing? No. That is actually the main finding or the main conclusion from Sharga's ratios. Second, species specific base composition? No, because AT and GC percentages can vary, or GC percentage rather, can vary from species to species many a times, and therefore there is some specificity in the base composition among different species that is maintained. Third, formation of the DNA double helix. So, of course, the DNA double helix stabilization is also addressed by the same. And fourth, the directionality of DNA strands. Yes, this is the fact that is not actually explained by Sharga's rules, and that would have been the correct answer. Next, in a DNA sample, A equals 30%, then what is the percentage of G? So A or adenine, if it composes 30%, then guanine would be composing how much percent? Again, a very simple math question relying on Sharga's rules. So if A is 30%, then G is also 30%. So that is taking up 60% of your DNA composition. What is left behind is 40%. So that gets split into two for G and C. Then G becomes 20%, then C becomes 20%. So the correct answer in this case would be 20%, which is a percentage of G. Next, which feature of DNA explains its ability to zoom in a little. Which feature of DNA explains its ability to replicate semi-conservatively? So, if you remember this, I don't think I've discussed the experiment in this case. I think it was Mendelssohn-Stahl's experiment that showed it. So, the semi-conservative mode of DNA replication is actually validated or is actually supported by which of these features? The sugar phosphate backbone, no. Major minor grooves, no. Complementary base pairing, possibly yes. And anti-parallel strands. So the correct answer in this case is complementary base pairing. So because there is complementarity between the two bases, therefore semi-conservative mode of DNA replication actually works. If there was no complementarity, then what would be the problem? Then you would not know which part of the DNA is like which part of one strand actually was base pairing with the with which part of the other strand, and therefore the sanctity or the correctness of the sequence would have been completely lost, and therefore complementary base pairing is instrumental for this purpose. Next, you have the correct order for chromatin organization and compaction. So, if you remember in our initial classes where we discussed the structure of DNA, we had discussed that your initial linear DNA forms nucleosomes, which then form chromatosomes. And the nucleosome is formed by the histone proteins, histone H2A, H2B, H3, and H4. And after DNA wraps around it 1.75 times on an average, following that, what you have is sort of like a stopper entity. The stopper entity is your histone H1, and that forms the chromatosome. And then the chromatosome also undergoes further coiling to sort of minimize entropy and maximize chromatin packaging efficiency, and that leads to the formation of the 30 nanometer fiber. So therefore, the correct option in that case is nucleosome. Sorry, this one. DNA, nucleosome, chromatosome, and finally the 30 nanometer fiber. Then the next question is that which level of gene regulation allows the fastest cellular response? Transcriptional, post-transcriptional, translational, post-translational. So the answer in this case is post-translational. Why so? Because the rest of the processes A, they require lot more external stimulus, and they are much more tightly regulated, and any shift in any of these processes can have very far-reaching effects. However, if you talk about only a post-translational modification where your RNA is ready, your DNA has undergone transcription, your RNA is ready, your RNA has undergone translation, and your protein as well is ready. So now your protein only needs to be activated or inactivated, and that simply can be controlled by a post-translational mark like phosphorylation, like simylation, acetylation, etc. So that is an immediate addition or removal of a small group, which can have very significant impact on the regulation of certain pathways. So the correct answer in this case is post-translational. Then DNA methylation in the gene promoter regions usually leads to which of the following? So we had discussed different aspects of DNA methylation, but when we specifically talk about the promoter region, what what is that expected to lead to when there is addition of methyl groups on the promoter side? Then that does not I mean, it does not directly inhibit, but it sort of impedes the binding of polymerases and other proteins that need to be recruited. And instead, what happens is that the transcription of that site or the expression of that site can often be compromised, and many a times it also uh, aids tighter packaging of that region, overall leading to a phenotype which can be affiliated with gene silencing when there is DNA methylation in the promoter region. It's a repressive mark, not an activating one. So that is the conclusion from that part. Then, which of the following traits do cancer cells typically show? A. Global hypermethylation, promoter hypomethylation of tumor suppressor, global hypomethylation with promoter hypomethylation, or no epigenetic alteration. So the correct answer is global hypomethylation, which means that globally there is a decrease in methylation mark in the DNA, so that more and more genes can be activated. More and more genes are transcriptionally active, which are oncogenic in nature, and the promoter hypermethylation of certain regions is necessary, mainly of tumor suppressive genes, so that there is reduced expression or shutdown of expression of these proteins, thereby aiding oncogenic survival. Which mutation is most likely to be lethal? A silent mutation, a missense mutation, nonsense mutation at the 5' end, or SNPs in introns. So it's actually option C, that is the nonsense mutation at the 5' end. Now, one might ask why the others can be lethal too. Well, actually, the question is yes, they can be, but um, what is the, like, say for example, if there's a silent mutations of course will not make much difference, but let's say there's a missense mutation, it is very probable that missense mutation is going to be very lethal if it is a an oncogenic mutation or a altering mutation which completely leads to non-functionality of a protein. That is certainly probable, and possibilities are endless in this context. But the question has asked you for the most likely option in this case, and it's actually of the presence of a nonsense mutation very close near the 5' end. Why? Because that is going to generate a truncated and almost non-existent protein, and that has a far more chance of being a lethal um, alteration. And yeah, keep in mind that both these possibilities exist, but but it's more probable that it can be nonsense mutation at the 5' end as the more likely, more probable option. Then, in question 11, we have been asked which property of the genetic code reduces the effect of mutations. So, if you remember, we had discussed the different attributes of mutations of the genetic code, universality, the degeneracy of the genetic code, non-overlapping major, commaless, etc. All of these attributes have been discussed. Now, to figure out which of them actually can reduce the effect of mutations, it is uh, it is actually the degeneracy of the genetic code. Why? Because even though if there was no degeneracy and each of these codons were actually going to call for a different type of charged tRNA and were going to code for a different amino acid, then not even a single change would have been tolerated in the sequence, and every single change would have led to some sort of a disastrous, could have led to a very disastrous outcome. However, that is not the case, and instead, a very large number of alterations and many a time, many a times tolerated because the same set of codons with one or two base changes um, can accommodate this, can compensate or can adjust with this change, and still incorporate the correct amino acid in that case. So the degeneracy helps in this purpose, and there's something else also which helps, which is called as bobble-based pairing, but that's not under the purview of what we are discussing here. But that is another addition as well. Then you have SNPs or single nucleotide polymorphisms. They differ from mutations because of which of the following. So they differ from mutations because SNPs always alter the protein coding sequence. Do they occur in coding regions only? They are present at 1%, approximately 1% frequency in the population or less than that. Uh, or they're always pathogenic. So the answer in this case is that their presence or their frequency in the population demarcates or distinguishes them from mutations. Then Sanger sequencing is limited in comparison to high throughput sequencing, primarily because of which of the following. Sanger sequencing, this is something I just discussed like a few minutes ago, where we saw that if you look at the contrast between Sanger sequencing and high throughput sequencing methods, then Sanger uses, I forgot to put this, yeah. Sanger uses can only sequence one fragment at a time, whereas high throughput sequencing can do a multiplexing of this experiment and a large number of sequences can be studied at the same time. So that was the answer. The Hershey-Chase experiment was repeated using a virus with an RNA genome. Which isotope would be the most appropriate to track the genetic material? So if we have the RNA genome, sorry, yeah, if we have the RNA genome, then in that case, using uridine, which would have made it the most unique marker for RNA, would have been the most appropriate isotope for tracking, and that is something which could have been done. So this is a hypothetical question, but you might get questions like this somewhere else as well. Which modification would most directly reduce nucleosome stability? So nucleosome stability is not something we have discussed directly as a topic, but if you essentially understand what does that mean, the stability of a nucleosome, it basically means that it's a 3D structure or the interactions which make a nucleosome complete, that would be disrupted or that would be lost. And most commonly that can occur because of histone acetylation, where you have an acetyl group, where the presence of an acetyl group and the negative charge which is associated with it can disrupt the DNA protein interaction and reduce nucleosomal stability. Enhancers, they differ from promoters. Why? Um, is it because they are always upstream? Is it because they function only in cis? Or is it because they are orientation difference or they can act at a distance? The answer is that they can act at a distance. So promoter needs to be right next to the gene because that is for recruitment recruitment and for sort of appointing the RNA polymerase that this is what you need to focus on and this is your next target. However, enhancers can be located way far upstream or way far downstream. They can involve 3D chromatin structural rearrangements, etc., all of which are eventually going to help enhance the expression of genes. So enhancers are very, very different from promoters. Now, mutation in which element would be actually quite likely to affect tissue-specific expression. So again, this is not something that we have directly discussed in the class, but tissue-specific expression can get disrupted by a large number of things. I think I have. Oh yeah, it's enhancer. Yeah, we have not discussed it, but yeah, the enhancer sequence because many times you would have tissue-specific enhancers which are present, um, and if there is a mutation in that enhancer, because that can be unique to the sort of gene, let me put it differently. The genome of all cells are the same. However, certain genes are better expressed or more preferably expressed in certain tissues and not expressed in other tissues. And while there are multiple factors that contribute to, sort of controlling this phenomenon or regulating this phenomenon, one of the major contributors are enhancers. So there are some tissue-specific enhancers which are present, which actually preferentially guide the RNA polymerase and to those sites and aid its expression at a much higher rate. So yeah, that's what the enhancer does. Now, which statement about histone methylation is true? Is it always active? Is it always activating? Is it always repressive? Is it context dependent? The answer is it is context dependent because histone methylation has both activating and repressive features, and depending upon which histone it is and where on which amino acid of that histone that modification or that methylation has taken place, there can be variation that is present. Next, promoter hypomethylation in cancer most commonly silences A. Oncogenes, B. Tumor suppressor genes, C. Housekeeping genes, or D. Ribosomal RNA or rRNA genes. So, promoter hypermethylation commonly can silence tumor suppressor genes. Why? Because tumor suppressors are most commonly expressed in normal cells, which maintains the sanctity of such cells. But in context of cancer, the hypermethylation of the promoter of tumor suppressor genes is actually a hallmark of cancer because that is repressing the the oncogenic signal and sort of making the cell more conducive to acquiring oncogenicity or becoming more cancerous. Now, which mutation is most likely due to replication slippage? So, base substitution, small insertion, deletion, or indels, transversion, or deamination. So, most commonly, it is replication slippage is going to lead to insertions or deletions, or the random insertion of a base, or the random exclusion or deletion of a base from a sequence. So that is what you will most commonly observe. Then which spontaneous mutation changes GC to 18 most frequently? So it is the deamination of a cytosine. Again, you can refer to the slide for this to your slides for this material. And following that, which observation would have contradicted the Watson-Crick model of DNA? So if A is not equal to T in some single-stranded viruses, that could have contradicted the Watson-Crick model of DNA because otherwise DNA shows antiparallel strands. GC-rich DNA has a high melting temperature. Agreed, that is because of hydrogen bonding. Uniform diameter of DNA, that's also meant, but A not equal to T in single-stranded DNA viruses would have contradicted this. A transcription factor gene is mutated, affecting the expression of multiple unlinked genes. So, one gene mutation is affecting the expression of multiple unlinked genes. What do you classify that mutation as? It is a transacting mutation because the mutation in gene X is affecting the expression of Y, Z, and W, which are not linked. When we say linked, we mean that their chromosomal locations are not such that their expression is under the control of some similar machinery, not under the control of the similar cis-acting elements. And as a result, the transacting nature of this mutation is attributed to the transcription factor. Which regulatory step do you think provides maximum amplification of gene expression? Maximum amplification would be allowed at translation, because once you have one RNA that can get translated hundreds and thousands of times to amount of um, increase the amount of protein that can be synthesized from the same, and so gene expression is amplified at a maximum in this case. Which feature of the genetic code ensures that mutations do not affect adjacent codons? So the genetic code, the non-overlapping nature of the genetic code ensures that if there is a mutation in one codon, that does not affect the adjacent one. The triplet nature is maintained such that every time the ribosome scans through, it is only taking one triplet codon into consideration and then moving past to the next. Yeah, these were the questions that were supposed to be a part of your original exam, but yeah, that is how it has proceeded. So I will stop sharing my screen now. I will share this material, the link for this course anyway, so you can attempt it at your own disposal whenever it's possible for you. And yeah, that pretty much sums up what I had planned for our class today. But anyway, moving forward, we will move on to understanding DNA protein interactions and techniques which are used to study them. We'll understand high throughput methods which are used for it. We'll understand what are the strategies in this context. We'll just overall cover all of these topics. So, yeah, that is pretty much it. We'll begin that from the next class. It will take 2 or 3 classes to sort of wrap that up. And yeah, we have completed, I think, 13 classes today, and we had a total of 25 weeks scheduled. So we'll have only 12 more classes in the upcoming weeks. And as I've already mentioned over email, it'll begin with we'll have to accommodate 2 classes a week because due to my own commitments, I have missed a couple of classes in between. So don't worry, we'll wrap up our classes. We'll have 6th, 10th, 13th, 17th, 20th. In this manner, we'll continue. We'll cover 5 classes, 7 classes in this month, and 5 classes in the next month, so that we'll be done with the course by before 20th March. So that is the plan. But anyway, thank you for joining. Have a good night. Bye bye.